Good morning, everyone. So I'm sorry I couldn't be there in person. The Pennsylvania chapter has been probably one of the earliest chapters to start the breeding program. And I really appreciate your support through this entire process over the last couple decades. So I'm here today primarily to talk about chestnut blight disease, which has killed 90% of the American chestnuts from the original, before the blight disease came through. So before the blight, there was approximately 4 billion trees. Today, there's 400 million. So that gives you kind of an idea of how severe this was, even relative to the current pandemic that's sweeping the globe for humans. And the backcross breeding was predicated on this idea that we could take the Chinese chestnut, it co-evolved with the blight, and cross it with American chestnut, and backcross out all of the genes from Chinese chestnut except for those involved in blight resistance. So the idea was that this is a simply inherited trait, and that at each generation, we could inoculate the trees with the chestnut blight fungus, inoculate these hybrids, select the ones that carried the resistance from Chinese chestnut, and basically continued the dilution process to get trees that grew like American chestnut but had the resistance of Chinese chestnut. So we're reaching the end of this original plan that was proposed by one of our founders, Dr. Charles Burnham. And what we do at the end when we get these third backcross trees, which are expected to be 95% American chestnut on average, is we cross the trees together so that their progeny have a chance of inheriting genes from both parents. And this is a numbers game. We have to generate hundreds of seeds, thousands of seeds from each of these crosses in order to make a few selections from each of the individual crosses. So we select currently about 1 out of 150 trees. And in our Meadowview breeding program, which has been going since the 1980s, we're now getting to the point where we're selecting the 1% of trees in these orchards. So in our seed orchards, we have 2 seed orchards at Meadowview, and we've planted 60,000 trees in both of those seed orchards. And one of them, we're down to about 500 trees. So that original orchard had 36,000 trees planted since 2002. And Eric Jenkins, one of the people that works at the farm, one of his mission statements, it was, "I plant trees and kill them." So we spend a lot of time planting, maintaining these orchards, inoculating them with the blight, and then we cut them down and we leave a few trees remaining to open pollinate one another so we can generate large quantities of seed for restoration. So the things I'm going to discuss in this talk is how do we measure blight resistance, and when we go through this selection process, and what are the relative effects of genetics on environment on this disease. Is blight resistance a simple trait that was originally proposed by Charles Burnham, or is it controlled by many genes? And what are the implications for breeding? And what do we know currently about the mechanisms of resistance to chestnut blight, and how can we learn more? And finally, what does success look like? We've been working on this for 35 years. Before the American Chestnut Foundation, there was already an effort starting in the 1920s to breed these hybrid trees. So we're talking about a multi-century long restoration effort process. So we want to be able to have some idea of where we're going. So for the first question, what are the relative effects of genetics and environment on resistance? For a lot of traits, actually, if we were to partition the variation in that trait, a lot of traits in trees, environment controls more than half of the variation. So a typical trait might be like 30% genetics, 10 to 30% genetics, and 70 to 90% environment. when we talk about the genetic component, it can be partitioned into the particular genes. How are there many genes that are controlling the resistance, or are there a few with large effect? And in some cases, we can't necessarily know. We can kind of map and statistically detect the larger effect genes, but then there's a lot of other genes in the genome that we can't actually see with our statistical methods, but we know that there are some total effect of small effect genes. And then when it comes to environment on chestnut blight disease, we have things like, where's the tree growing in the plantation? The edge of the plantation, what I've noticed is the trees that are growing on the edge, they have a lot of resources. They tend to look a lot better than the trees that are competing with each other in the middle of a plantation. We have, how do we measure the resistance itself? Some bias in that. The actual site, is it good for growing chestnuts? If it's very wet, the trees don't grow as well as, or they have more disease than if it's a drier, well-drained site. So all of these play into this complex mixture of what actually what we see as the phenotype of the tree, which is the resistance. So in the process of doing selection in our seed orchards, the first step is to inoculate the trees with a weakly pathogenic strain of the chestnut blight fungus. And we inoculate the trees around age 2. We see this spectrum of canker disease severity that occurs, and we call out trees that demonstrate significant canker expansion. So in the figure, ratings 2 and 3, a lot of those trees we remove from the orchards. So right off the bat, we are doing a pretty heavy selection step, and we're left with 10 to 20% of the trees that we initially planted to make selections of the 1%. So then we have another step of, as this disease progresses, it has some additional phenotypes that develop that you don't see necessarily in the first 6 months after inoculation. And that is, the main stem survive? Does the main stem survive over, 5 years plus after the inoculation? Do the trees have large cankers? What we do, just a quick field assay, is the canker larger than the size of your palm? Are the cankers sunken? If they're sunken, then typically that means that the blight is penetrating into the vascular tissue of the tree and it's not doing a very good job of fighting off the fungus. Whereas more of a swollen cankers indicate that there's a walling off of the infection. Does the cankers eat through the bark and expose the wood? Is the fungus sporulating or not? So all of these things kind of give us an idea, a gestalt of how resistant the trees are. And just looking at these phenotypes themselves, what I have in this figure is, what is the percentage of variation on the table on the left? Is what is the percentage of the variation that's explained by genetics, which is the heritability. And for many of these traits, genetics only explains about 10% or less of the variation. So on the figure on the right in this thing is looking at the probability of the phenotype that we observe given the tree's genes. And what the box plots are showing is that probability is kind of overlapping. If you observe a main stem or not a main stem alive, what is the probability that that tree, if you were to plant it out, many copies of that tree, what's the probability that the stem would survive? And you see this overlap in these phenotypic classes, indicating that there's a kind of weak genetic signal. So we can't always judge a tree by how it looks. And what I've done for looking at the overall resistance of these trees, I've built up selection indices, basically the sum of all of these probabilities for all of the traits to rate individual trees. But just looking at those individual tree phenotypes is maybe not good enough to make selection decisions. So we also want to screen the progeny of the trees for resistance, because that gives us an idea of the genetic component of the resistance. We can see, we basically go through that seed orchard, we partially select it, we're left with 10 to 20 trees in a plot of 150. We allow those trees to open pollinate one another, and we plant their progeny out in randomized field trials. We inoculate the progeny with the chestnut blight fungus. We look at the canker expansion, and then we kind of judge the parents based on the severity of the cankers of their progeny. You can imagine this is a very time-consuming process. We have to wait for the trees to grow up and flower. So it typically takes about 7 years for a tree to, from the time we plant the seed to when it actually flowers. And not all trees flower at the same time. So we, out of like thousands of candidate trees that we have left in our seed orchards, we have progeny tested 1,000 in the last decade. So it's too slow to actually make final decisions. When we look at the heritability of canker severity on the progeny, we're looking at multiple progeny per tree, so that gives us a better estimate of the genetic component of resistance. When you look at multiple progeny, we see that the heritability, about 60 or 67% of the variation is explained by the parent tree. So that's giving us a more accurate estimate of the resistance. And what we're doing for looking at resistance is we're taking the length of the cankers, and we're also doing the subjective ratings, and we're summing these together to get a canker severity estimate for each tree. And then we're taking the averages, essentially, of those canker severity measurements for all of the progeny of a given mother tree. So to accelerate this process somewhat, rather than planting trees in the field and waiting 3 years and having a lot of the trees just die for other reasons besides the blight, so we end up losing progeny before we actually end up assessing them for blight resistance, we've been shifting towards collecting seeds from the mother trees, planting them up in a greenhouse, and inoculating seedlings and evaluating the blight resistance. And what we do is we measure, for the blight resistance, we're measuring primarily how long do these seedlings survive. So that's the days to wilt. And at the end of about 5 or 6 months, we look at how many trees survived within a given family. And basically what we find is this, how many trees survive is the most accurate estimate of resistance at the end of these trials. And we do end up killing 90 to 95% of the trees at the end of these things. So it does not necessarily, it's not a good assay for trying to keep trees alive and selecting, but it does help speed up the screening of progeny. So even with this rapid screening in a greenhouse, we still are limited in the rate that we can screen progeny from trees, and it's a massive effort. So one thing we've been doing is, for all of the mother trees that are left in our seed orchards, we've been genotyping them. We've been taking leaves, extracting DNA, and looking at what are the DNA variants in that population. And we are correlating the DNA with the variation in resistance in the population. So we've evaluated the parent resistance, we've evaluated the progeny in the field, and we've evaluated trees with small stem assays. So currently we've done genotyping on over 3,000 selection candidates from our seed orchards, and we've developed those prediction models for parent trees that we haven't screened progeny for, or maybe are too young to assess this long-term resistance. So we're able to get resistance predictions for all of our trees so we can compare them all on the same scale. And so just kind of emphasizing this is that we've done the phenotyping and genotyping on different subsets of trees. And what the genomic selection enables us to do is put everything into one scale. And so we can just put all of the trees in one big spreadsheet and say, this tree has higher resistance than this tree, and we can rank the trees and go ahead with our selection. So how does this work? Basically, what we're doing with genomic selection is we are looking at how closely a tree that we don't know the phenotype, we don't know the resistance from the progeny testing, is to trees that we do know the resistance from progeny testing. So what genomics adds is we have this very fine scale idea of how closely related trees are to one another. And that gives us a more accurate picture of how resistant the trees will be that we have not actually screened progeny from. And so, you know, with your siblings, we expect that you would share 50% of your genome with your siblings. But with genomics, we find out that you might share 35% of your genome with one of your siblings and 65% with another siblings. And so the genomics helps give this fine scale relatedness, which then gives us more accurate estimate of prediction of, let's say, your height relative to your sibling's height, for example. Same thing, it works the same way in trees. And just looking at how this relates back to our BC3F2 populations, we have pedigree relationships, which are the strength of relationships between trees in our population based on just what we know about who is their brother and sister, who are their parents. And that's on the y-axis here, and then the genomic relationships on the x-axis. And you see that while what we expect from just the pedigree of the trees is correlated with the genomic relationships, we see that the genomic relationships gives us a lot of spread, a lot of deviation from what we expect. And that gives us this fine scale ability to predict resistance. So now when we do these predictions, looking at the various ways that we measure resistance, we have progeny resistance and parent resistance, and that's over on the left. And you can see that they're correlated, so that's good. We are actually measuring resistance with both of these measures of parent and progeny, and they're correlated with one another. Now, when we look at small stem assays relative to these parent resistance or progeny resistance, we see weaker correlations. However, they are still positively correlated. So that indicates that the small stem assays may not be as accurate of a measure of these trees' resistance as opposed to putting progeny out or evaluating the long-term resistance of the trees. But when I actually went to make the selections, what I did is I just summed the parent resistance, the progeny resistance. I put a little bit more weight on, 3/4 progeny resistance from the field, and then 1/4 small stem assay resistance. And so that gives us the blight selection index. We're looking at this multi-generational ability to resist blight. And in the figure, the red dots are our selected trees. The black dots are the trees that we're going to call out of the population. And the purple dots are trees that are F1s, basically. So they're hybrids between Chinese and American chestnut. So we see that our trees on average are somewhere around F1 resistance or a little bit less. So what do these trees look like? They have an intermediate spectrum of intermediate blight resistance. So you look at American chestnut, it gets the blight, it dies back, and it resprouts over and over again. Our BC3F2 hybrids, these trees are on average about 85% American chestnut, one of the ones that we've selected, and they have the main stem often does survive with the blight infection. They get blight fairly severely up and down the stem, but there is a sign that the tree is walling off that infection. Now, when you look at the trees that were F1s, basically what happened was when we were generating these trees, these BC3F2s, we were growing the third backcross trees in an orchard environment with Chinese trees. And the Chinese trees sometimes pollinated the third backcross trees. And so we ended up with F1s on accident in our orchard, and they actually ended up serving as a kind of control to see like how good are the backcross trees that have more than 50% American chestnut ancestry relative to these F1s. And we see that on average they're not as good as the F1s. So we are in this gray area of, yes, our breeding populations have resistance, but we don't know if we have enough resistance for restoration. So getting back to this idea that the whole program was based on the idea that blight resistance was controlled by few genes. And if it was controlled by few genes, then what you would expect is as you increase the amount of American chestnut ancestry through backcrossing, you should have this spectrum of blight resistance, and trees that are near 100% American chestnut ancestry should have the full resistance of Chinese chestnut. That's pictured on the left. If you have many genes involved, as you do more backcrossing to American chestnut, you lose resistance along at every generation. And so we can now finally, with the genomic data, kind of evaluate this hypothesis. We looked at American chestnut ancestry relative to resistance, which is based on all of the things I just talked about. And we find that there is a strong trade-off. We do have a few backcross trees that have resistance similar to the F1 hybrids. And there is quite a lot of variation in resistance within a given American chestnut ancestry. So that does give us the ability to select for resistance within a particular amount of American chestnut ancestry. However, we are not going to get trees, no matter how much recurrent selection or anything that we do in the future, we're not going to get trees that have 100% of the resistance of Chinese chestnut and have most of the American traits. We're falling within this range of intermediate resistance and intermediate traits. So we can also now look at which regions of the genome are associated with resistance. How many are there? What is the effect of these individual regions of the genome on resistance traits? And basically what I've worked with some collaborators to look at which regions of the genome of these backcross trees were inherited from Chinese versus American chestnut. So we can look at this mosaic of inheritance on all of the chestnut's chromosomes and look at how does, if a tree inherited Chinese genome or American chestnut genome at a particular part of the genome, how is that correlated with resistance? And we scan the entire genome and we find regions that are associated with resistance this way. And just looking at, this is a complex figure, but what we're looking at is there's 12 chromosomes in chestnut, and these are all traits going up and down on this figure. Main stem alive, large cankers, sporulation, exposed wood, some cankers, and all of the traits summed together. And the lines on this figure are indicating which parts of the chromosome are correlated with resistance. And we have two sources of resistance from two different Chinese trees, primarily in our breeding program, the Clapper and Graves sources of resistance, which you're probably familiar with. And what we find overall is that about 1/7 of the genome length of these hybrid genomes is associated with resistance. These regions represent, each of these regions that are associated with resistance encompass about 6 million DNA bases. So we're not actually getting down to the individual genes in these regions. The average amount of variation explained by a single peak on this graph is about 3%. So that means that these individual genes or individual clusters of genes each have small effects. And then what we're detecting is not actually the entire story. So the total trait variance explained by all of the regions that are associated with resistance is somewhere between 2% and 57%, depending on the trait. So the largest single QTL, or the region that's associated with resistance, is 13%. So what we see also is Clapper and Graves, our two sources of resistance, have both unique and shared regions that are associated with resistance. So you might think, okay, Clapper and Graves inherited different genes. So what if we cross them together, putting so we can get both genes from Clapper and Graves in the same trees. And we actually have, in a way, done this already, where we've had our BC3 trees in the same field. The Clapper and Graves trees at Meadowview were planted basically on one side of the road and the other side of the road. And with DNA, we're able to tell, based on the genotyping of the BC3F2s and the BC3 parents, which were crosses between Clapper and Graves and which were crosses within Clapper and Graves. And the crosses between, if you think that they inherited different genes and crossing them together, you would see an enhanced amount of resistance. And what we see in this figure is that basically American or Chinese chestnut ancestry is driving the resistance. That's what's the primary source of variation. Crossing between the two sources of resistance doesn't really have a strong effect. So basically, what are we going to do going forward with this kind of middle of the road resistance that we're seeing? I think one way to improve resistance is by increasing Chinese chestnut ancestry. And we want to do this as much as necessary and as little as possible. So what I'm proposing with the chapters is that we make some earlier we cross some of these trees that have inferior resistance with, let's say, F1s or BC1 trees that have a little more Chinese ancestry. We incorporate those into our existing seed orchards, and we evaluate over a long term their resistance relative to trees that have more American traits but less resistance. Now, we're fortunate that the transgenic tree is potentially going to get through this regulatory process, and we could cross some of these backcross lines with inferior resistance with oxalate oxidase so that we can rescue those trees' genetic diversity so that your efforts in creating them are not lost. And I think overall we need to moderate expectations, both on the side of backcrossing, where we have to find a balance between this American ancestry and resistance, and it's really hard to determine that balance because it takes a long time for these trees to grow and we have to put them out in field trials. So right now we have uncertainty, so we want to be able to have multiple levels of ancestry and put those out in the forest and see how they do. And even with the OxO tree, the transgenic tree, we haven't evaluated that tree's resistance over the long term. So we still need an insurance policy with a breeding program to make sure that if the OxO resistance is not as good as we think, that we have some other pathway going forward for restoration. Now, finding a balance between blight resistance and American traits, what I propose we do is increase stringency of selection at the 3rd and 4th backcross stage, and that's what we are doing we're genotyping all of your chapter selections right now. We have a lot of data on Pennsylvania trees now at this point, and there's some additional genotyping coming down the pipeline. And also going back a little bit and intercrossing our third BC3F2s with F1s or BC1s and putting those trees in our seed orchards so that we have some lines that have increased resistance. So putting those earlier generation hybrids, one idea that was proposed by Paul Sisko, a former geneticist with the American Chestnut Foundation, was to pollinate American female trees, using Americans as females and Chinese trees as males. You create F1s that are male sterile because there's some kind of genetic incompatibility between the American chestnut chloroplasts and mitochondria, which are inherited from the female, and something in the Chinese nuclear genome. So what you get is trees that these male sterile F1s do not put out pollen. So if you plant them in an orchard, they will be pollinated by the neighboring trees, but they will not pollinate the neighboring trees. So we could collect seeds from them and plant what are called better BC1s, or basically trees that are around 75% American chestnut in their ancestry, but they inherited some resistance from these other neighboring BC3F2s. So that enables us to not, I guess, contaminate our orchards with lots of Chinese chestnut traits, but also selectively take some trees that have more resistance and plant those trees out in the orchard or out in the restoration trials. And then increasing stringency of selection for resistance. Basically what the regional coordinators are working on, maybe not this year, depending on the quarantines, how long they last, but what they've been doing is going out to all of the backcross trees that all of the chapters have generated through their programs, and they are collecting leaves, and so we can do genotyping and infer the ancestry of these trees, and they're also phenotyping them for the long-term blight resistance. And given the trade-off between American chestnut ancestry and resistance, I'm making a guess that trees with greater than 95% American chestnut ancestry are not worth pursuing in terms of doing continued intercrossing and selection among their progeny for the traditional breeding program. And actually, of the 521 candidate backcross trees that from the American Chestnut or the chapter genotyping project that we've genotyped thus far, about half of them have 95% or more American chestnut ancestry. So these trees that have potentially inferior resistance, we can cross with OxO pollen, and we're going to get trees that we basically have ready-made germplasm conservation, and these trees are already flowering and ready to go. Or we could make some crosses with earlier generation hybrids to increase the resistance of the progeny. So outcrossing transgenic blight, once we have, if this OxO tree gets through the regulatory process, this is a very efficient method of rescuing genetic diversity, is taking that OxO pollen, applying it to wild type trees or backcross trees that are mostly American in their ancestry. 50% of the progeny will inherit the gene. And you can see the stark difference in resistance between trees that inherited the gene and the trees that did not inherit the gene. So this is a lot easier in terms of the numbers that we have to generate. So it's operationally more feasible for a citizen science program to undertake. And we can't simply take this transgenic tree and cross it over one generation to wild type trees and put out the progeny and expect that we're going to have a restoration product, because we're starting with a single founder tree and crossing it to even thousands of wild type trees. We still have 50% of the genome of these trees that are inherited from one tree. And trees tend to carry a lot of deleterious bad mutations in their genome. So if we were to just stop at one generation of crossing, what we would have is the trees that start intercrossing, and if there's any bad mutations in the original founder tree's genome, they would become homozygous when we cross the trees together, and then the trees would be less fit for restoration. So what I had proposed was doing 3 to 5 generations of outcrossing to wild type trees with increasing numbers of individuals in each generation. So that gives us time to find wild type trees, get them conserved. We don't have to do as many crosses. And we're basically diluting out that founder tree's genome, and we're increasing the amount of diversity that's represented from the wild tree at a feasible number of crosses we have to make, which is about 675 crosses over the next decade or so. So that will get us to a population that is hopefully able to be diverse and resilient to climate change and represent the diversity that's remaining in the species. And we've already started doing this. We under permit from the USDA. We've had in 2019, SUNY made crosses onto 20 wild type trees. Meadowview made crosses onto 3 large surviving American chestnuts and 2 of our better hybrids. And Purdue also made crosses onto 10 wild type trees. And we generated over about 1,000 seeds from these crosses that tested positive for the inheritance of the oxalate oxidase gene. And this is these pictures are showing how we do the tests, which are fairly simple. We take each nut and we put it in a bag. We have an individual ID for each of those nuts. We take a core from the nuts before we even plant them. We put them in a solution that changes color if the tree inherited the gene or not. So it's a lot more straightforward. We can know exactly, even before we plant the trees, which trees inherited the genes, and we can plant out the ones that inherited the genes for future crossing. So what do we know? I mean, taking a step back, what do we actually know about the biology of blight resistance and the infection? And basically, what happens with a chestnut blight fungus is it grows into the bark, that has the mycelium grow into the bark, they cause necrosis, they kill the living tissue in the bark, and then they girdle the stem. So we know from Bill Powell's work that oxalate, the secretion of oxalic acid by the fungus is an important mechanism of virulence, and therefore detoxifying that acid is important for resistance. And what he found also interestingly is that Chinese chestnut has a mechanism of detoxifying this oxalic acid. So he's inserted a gene from wheat that detoxifies oxalic acid, but Chinese chestnut already has this detoxifying mechanism. And Chinese chestnut, it appears they don't have a single oxalate oxidase gene, but instead they have a pathway involving multiple enzymes that are involved in detoxifying the acid. Whereas American chestnut is very susceptible, and these leaf discs are American chestnut and Chinese chestnut and transgenic trees that were soaked with oxalic acid. The brown is necrosis as a result of the oxalic acid killing the tissue. But that's not it's not as simple as that. I mean, we've seen huge parts of the genome that are associated with resistance. So it's not just that there's lots of oxalic acid detoxifying genes at many locations in the genome. Fred Hebert, a long time ago in the 1980s, was looking at comparing Chinese chestnut and American chestnut bark that was infested by chestnut blight. And he found basically that the mycelial fans that form and grow into the wood form more rapidly in American chestnut relative to Chinese chestnut, which is resistant to the blight. So what is happening to at a metabolic level in the cells of that bark to inhibit the growth of those fungi? We don't know. He also found that the rate and thickness, what happens over time is that the tree recognizes that it's being invaded by the chestnut blight fungus, and then it starts forming a wall around the infection by forming, the cell walls are thickened with lignin. It forms this waxy surface on the cell walls, and the fungus is walled off. And the rate that occurs and the thickness of the walling off the infection is an important part of the resistance mechanism over the long term. And then the tree is able to slough off that infection as the bark grows. So there's a reprogramming of cells towards forming lignin that somehow the fungus or the tree recognizes that's being invaded, and then it starts making reprogramming cells. So we want to discover additional mechanisms of resistance besides just oxalate detoxification. And one of the ways that we're doing this is sequencing the Chinese chestnut genome and the American chestnut genome with the latest technology. The Chinese chestnut genome has already been sequenced at Penn State with John Carlson's lab. What we're working now with Hudson Alpha Institute for Biotechnology, which is in Huntsville, Alabama, and they are using this new DNA sequencing technology, which enables us to build very high quality reference genomes, basically that have all of the DNA assembled nicely into chromosome level assemblies. So we can see which genes are present and compare the Chinese chestnut and American chestnut genome. I've already also showed you this, but we're scanning these hybrid genomes for regions correlated with resistance. We get huge parts of the chromosomes that are associated with resistance. It's very hard from these data alone to identify underlying genes. So we need another layer of information, and that is looking at gene expression in the stems of Chinese chestnut and American chestnut after blight infection, also looking at metabolites, proteins, whatever additional layers we can add on to that, we can see which genes are more highly expressed in Chinese chestnut quicker than American chestnut, and are they located in these regions associated with resistance? Then we start narrowing down to candidate genes. And actually this, there's already been work done on this with the Forest Health Initiative that was funded almost a decade ago, where there was earlier iterations of this genome assembly and gene expression analyses and mapping of resistance genes, and they identified a number of candidate genes, about 25 or so they put into American chestnut from Chinese chestnut, and now we're just getting to the point of assessing the resistance, and this is data from trials planted near Virginia Tech from candidate genes that were inserted by Scott Merkel's lab at UGA and Bill Powell's lab at SUNY ESF. And these are all canker or susceptibility to blight, just looking in the field. Lower numbers are better here, and all of the bars are different candidate genes. And the far on the right is wild type American chestnut. So you can see from preliminary data, we seem to see that some of these genes have a partial, they partially increase resistance. So beyond having this oxalate oxidase gene, we also have some additional genes that we could put into Chinese chestnut that might give more robust resistance. Now, thinking forward to CRISPR, it's getting to the point now where CRISPR used to be where you could only do kind of knock out genes, like delete genes or reduce their function. Now you can actually insert genes in a targeted way with CRISPR. And so one potential application using CRISPR going forward is, let's say, putting the oxalate oxidase gene next to another one of these Chinese chestnut cis genes, next to a gene that we identify for root rot resistance. So basically we have one, we've taken this complex polygenic resistance, many genes involved, and simplified it down to a single locus. So we could essentially select it as if it is a single locus, and it's much easier to do breeding that way than having to select thousands of progeny and then do selections on multiple loci at the same time. The other potential application of CRISPR is to get around this founder bottleneck. So with the oxalate oxidase gene, it's been put into a single tree. And if we could insert that gene into the same location in multiple founder trees, then we reduce that founder bottleneck significantly. And let's say we could do that for two, it would, even just 2 trees, it would improve the size of that population by double. So what we do early on has a profound effect later on for the amount of diversity that we've captured in the program. So why we could do that now with agrobacterium insertion, but the gene gets inserted in random locations with agrobacterium, and if we have a lot of copies of the gene at different locations in the genome, what happens is as these trees cross together, they end up inherited in multiple copies, and then the cells sometimes shut that expression of that gene down. So we want to avoid that process of gene silencing, it's called. So what does success look like? Looking, zooming back out to like, what is our big picture goal? And I think there's about 5 criteria we could say would constitute success. And that is the main stems survive indefinitely with blight infection. Trees grow at rates and maximum heights more similar to Chinese chestnut or American chestnut than Chinese chestnut. So these trees are competing as canopy trees. We want to have the iconic look of the American chestnut, the leaf, twig, and nut characteristics, similar if not indistinguishable. We want to represent the diversity that's left after the blight. so that these trees can continue to adapt to climate change. And we want to have a subset of the population that has resistance to chestnut blight and the second pathogen, Phytophthora root rot. So now comparing breeding and biotech with these different approaches, I think we can get the main stems to survive indefinitely if we increase the Chinese chestnut ancestry. And biotech, yes, it looks like we have high levels of resistance with the oxalate oxidase gene, but it's too early to assess the durable resistance. For sure, biotech, because they're only inserting a single gene, and that gene does not affect the overall metabolism of the tree, besides, in fact, increasing its resistance, these trees are going to grow and look like American chestnut. And whereas breeding, the Chinese trees have a lot of genes that make them grow more like orchard trees, so we may not get to that high height growth with the breeding, and that might be a risk with increasing the Chinese chestnut ancestry. We represented the diversity in the population? We may have in our backcross program, but we still need to compare the diversity of our backcross populations to wild populations, and we're doing that now. And we will certainly represent that diversity if we outcross the biotech trees to wild populations. And a subset of our population in the backcross program has resistance to Phytophthora. So if we bred the Phytophthora resistant trees with the transgenic tree, that's one way to combine resistance to both pathogens. So that's what we're planning on doing, is we're taking our backcross trees, we're selecting them for Phytophthora resistance rather than blight resistance. We're planting the survivors out at sites where Phytophthora is present in the soils, so we're not introducing Phytophthora to new locations. And eventually we're hoping to outcross those Phytophthora resistant selections with blight resistant transgenic trees. So this will take another couple generations of breeding to get trees with resistance to both pathogens. And it is possible to generate backcross trees with high levels of American chestnut ancestry and high levels of Phytophthora resistance. This figure is showing, ancestry versus resistance. We've made some Phytophthora resistant selections in our MetaView breeding program with genomic selection, and we are able to generate these highly resistant, highly American selections. And that indicates that there's likely few genes compared to blight resistance that are having a major influence on resistance. And we actually map where those genes are in the genome for resistance to Phytophthora. We see one chromosome essentially with two regions on that chromosome that have major effects. Those regions don't explain 100% of the variation, but this is more promising in terms of a trait that we can do backcrossing to generate resistance. And we're also in the chapters, I want you all to continue to find, as this quarantine passes, continue to find wild trees and mark where they are in TreeSnap, this app that you can get for your phone. We want to be able to go back to those trees in the winter and collect scion wood from them. And currently we are on track. We're trying to conserve about 100 different sources of wild trees this season. And it looks like before this quarantines occurred, a lot of our regional coordinators and chapter members have essentially reached those targets. And we are planning on grafting some of those scion to rootstock this year if it's possible. Of course, if you find rare flowering trees, we really want to know about them so we can collect seeds, because that's ideal. Because grafting, the trees are susceptible to dying from the blight infection at the graft union. So we'll be continuing to ramp up our germplasm conservation efforts in the next few years. So looking at the big picture of how we're doing this, we've diversified from just doing backcross breeding for blight resistance. Now we're selecting for Phytophthora resistance in our program. We are taking our backcross lines that don't have a lot of blight resistance, and we're putting them into germplasm conservation so that we could eventually cross them with transgenic trees. We're using genomics to inform all efforts, all parts of the effort, including, where's the diversity, what's important to conserve, how can we speed up the selection, how can we discover genes for resistance, and eventually put them into American chestnut to bolster resistance beyond what we can achieve with just the oxalate oxidase gene. So with this multifaceted approach, we increase our chances of success, and we need to have multiple approaches given the uncertainty of how long it takes to assess resistance in our success. So that's all I have to say. Thanks for listening.
Jared, thanks so much for joining us remotely. Excellent presentation, as always. So we've got a couple questions. Before I get those, I just want to remind folks, if you have a question, please use the chat box, the Q&A, email Hoy, text me. I'm also opening up the poll. So I've got a poll about this, this webinar process. We'd like to know how you liked it, if you want us to do more of these. Kind of a personal question, would you have preferred to meet in person? Because I know I would have. I'd love to have seen a lot of you guys whom I don't get to see very often. So it's only a 4-question poll. So please answer that if you get a chance in the Zoom format. So Jared, I've got a couple questions while folks are maybe answering additional ones. The first, Louise asks, how about incorporating Japanese chestnut into the breeding program?
Yeah, I mean, I think we have somewhat, even in the Pennsylvania, let's say the F1, F2, F3 program, there are Japanese chestnut parents represented there. And we could potentially, if you find timber type Japanese chestnuts or F1 hybrids derived from Japanese chestnuts, we can incorporate them. One thing that I would like to pursue in the future is even stepping back to looking at fundamental mechanisms of resistance in both Chinese chestnut and Japanese chestnut, which would entail getting a range-wide collection of these species, planting them in a common garden, inoculating them with the fungus, sequencing the genomes, and seeing if there's variation in those species that we can map genes for resistance. So I think we need to go back to the fundamentals of trying to understand basic mechanisms of resistance to enable new biotechnology in the future.
Thanks, Jared. So I've got, let's see. Clark asks if we're taking lunch recommendations. I know that we're having grilled cheese and soup here with the kids. Let's see. Oh, Carl Gallegos asked about studies that have been done with mycorrhizae and American chestnut, and I think his question was most curious because I know there have been plenty of studies done on American chestnut and mycorrhizae. Do you know of any that have been linked toward helping chestnut to adapt to different types of environment, maybe because of climate change or just to try and extend their range of growth capacity?
What I can recall from just reading some scanning literature on mycorrhizae is that it does look like there are some unique associations of mycorrhizal fungi with American chestnut and versus like hybrids and Chinese chestnut. And I think that mycorrhizal associations and just generally the vigor of the trees does interact with blight resistance. So we have done, Sarah, I know, do you guys actually do some mycorrhizal inoculations of the soil? I know Kendra does that to improve some greenhouse growth conditions, but we haven't done that a lot in our orchards. So I think that generally though, for restoration, if we can find sites that are well suited to American chestnut that will grow very well, we can have trees with intermediate resistance do very well on the best chestnut sites, which probably had chestnut before and has those mycorrhizal associations that help the chestnut be robust, grow well on those particular sites.
And then just to add a couple of the studies I know, from minelands, reclaimed mineland studies, those I think are some of the more interesting ones because minelands are typically reclaimed mineland sites are usually pretty sterile. They don't typically have mycorrhizae in the soils. What they found is Usually you get the beneficial mycorrhizae coming in about 3 to 5 years after planting, and they help. So mycorrhizae certainly help with establishment. They certainly help with growth. They can do good. It doesn't appear to be necessary for all sites because you can get mycorrhizae coming in at a later date. But I would say if you have a marginal site or if you have a poor site, I would certainly recommend the application of mycorrhizae because they will certainly help with the establishment and growth. But if you have a good site already, I wouldn't worry about it.
Yeah, the other thing to think about is like the Phytophthora. Just having competing fungi with Phytophthora, you can have Phytophthora present in the soil and it will not cause disease if there are lots of other competing fungi and things that will protect the roots. So having, inoculating mineland soils that are fairly sterile with mycorrhizae could help ameliorate the Phytophthora problem.
Great. All right. Let's see. Another question. Lake Roski asks, why does this is a long question, so bear with me. Why does having more genes make it impossible to get 95% Castanea dentata with required resistance? Isn't it just a matter of increasing the number of selections and going through many more generations of selections? It's like you're saying it's not possible to move the required Chinese resistance genes onto the American genome without dragging much more Chinese genes. Why doesn't additional selection and breeding, for example, crossing Clapper and Graves with new sources of resistance, eventually lead to an American type work? People are saying that backcross breeding will not work no matter what.
Yeah, I mean, the challenge with that is that let's say we had perfect knowledge of all of the genes for resistance, which we do not. We could design perfect DNA markers and we could plant out thousands and thousands of progeny and assess all of them for whether or not a tree inherited those genes. And then we could make selection for more and more American type around the genes so that we don't drag on these additional genes from Chinese chestnut. But the problem, what makes it so challenging is that measuring resistance is not straightforward at all. A lot of it's environmentally dictated, so we are not making perfect selections and we don't know all the genes involved in resistance. So yes, we can continue with recurrent selection and select for more American type, but even like measuring the height growth and things like that, super hard to measure because you're waiting for decades to see like when does the apical meristem stop being dominant. So it's just the challenge is our lack of knowledge about what are the underlying genes and having a perfect selection mechanism for actually accurately selecting for them in every generation. That's why it's so challenging.
All right. Let's see. So from Larry Grossman, have you been able to genotype longer surviving Native American chestnuts identified through programs like ACCF?
We really want to do that. I'm super excited about that, Larry. So what I've seen in at Meadowview is Fred Hubbard and others have done crosses between these large surviving trees and planted them at Meadowview, and they do have really nice resistance, some of them do. And so what we're planning on doing is sequencing the whole genomes of those wild or the large surviving trees, including the oak tree, for example, in Pennsylvania, and then comparing those wild trees or the large surviving trees with some resistance to trees, American chestnut trees that we know are susceptible, and looking for systematic differences between the DNA of the large surviving trees versus the susceptible trees. So I really do want to like map resistance mechanisms in the wild large surviving trees. And then the other way we're doing that is we have graft propagated some of these large surviving trees and graft propagated some susceptible trees, and we're inoculating them with the chestnut blight fungus, and we're going to compare gene expression in the stems after inoculation in large surviving and susceptible trees. So we're doing kind of similar approaches of mapping and understanding resistance in the wild trees. And it would be really nice to be able to, let's say, do CRISPR on the susceptible trees to enhance the expression of genes that might be involved in resistance in the American chestnut.
All right, thanks, thanks, Jared. And I also got a thanks so much from Lake for the answer that you gave. I've got two more questions, one comment. So I don't know if you guys saw, but Mark McDill came and gave an excellent post in here, a response on whether American chestnut is appropriate for future climate in PA, and it looks like yes, and I would agree with that. And he gave a link there. The other is a response to the Martian that Ethan Habriel posed earlier. He put another good link in about giving some additional information to that. So Ethan, I'd recommend checking out that link if you get a chance. So the last question that I have is from Dave Laser. Jared, I don't know how familiar you are with the Smith Farm that's in Jefferson County here in Pennsylvania, but he wondered how might those orchards or farms be best incorporated into the plan that you've outlined.
I'm not familiar, Sarah, so I'm going to leave that up to you to answer.
Yeah, so you know, Dave, you and I have talked about this a couple of times. You know, the reclaimed or the mine lands that are on there, continuing that mine land research is certainly very interesting and compelling and important to the work that we're doing. Jared, I don't know, you don't know this, but Dave has done some pretty interesting plantings. There are some trees planted in the reclaimed mineland area, and then trees planted in an unclaimed or in an unmined area, and you can see the huge differences in growth, as we would expect. But it's one of the few places where I think you've got plantings in both mined area and unmined area so close together. And those have been compelling. As far as what to do to continue, Dave, I think you and I can have a powwow and maybe we can rope Jared into that in the future. But there are certainly things that we can do there and continue. You and I have talked about doing pollinations of local trees with the transgenic tree, or maybe doing plantings of transgenics there as well. We also still want to try and rescue some of the backcross trees that are there. So I think there's things that we can do there in the future.
Are there backcross trees that could be genotypes there?
Yeah, but we haven't been able to do selection because they were planted in the reclaimed area before we really knew how to reclaim mines. They're super stunted. And so we have not done any selections. So maybe, but it won't be in response to inoculation, I don't think. I think they're just too stressed to do that. So let's see. We've got a couple of thank yous, and I don't see any additional questions. So thanks, Jared. I'll talk to you in another webinar very soon.